So in my last video, I was talking about how aspartate and ammonia was able to be made in the liver, and those are the substrates that we're going to be using in the urea cycle. So if you could think about these nitrogens all coming in and aspartate coming in, we're going to start our urea cycle. So our urea cycle, very important, is that they utilize two main substrates, and then that's going to be ammonia. I should put ammonia and aspartate. So one of the major points of the urea cycle is that it happens in two places, in two compartments of a cell, and one of them is going to be the mitochondria, and the other one's going to be the cytosol. So I'm going to just put a little wall over here, and I'll just say that everything on the left is the mitochondria, and everything in the cytosol is going to be on the right. So the urea cycle invariably starts from the mitochondria. And what we're going to start off with is we're going to start off with our ammonia. And it turns out that the first step of the urea cycle is in fact the regulated step. What it combines is bicarbonate with ammonia, and using this highly regulated enzyme, which you have to know, it's going to be carbamoyl phosphate synthase. It's abbreviated many times as CPS. This will make a carbamoyl phosphate. So this is one out of two carbamoyl phosphates that you will learn, and the second one is going to be in pyrimidine synthesis, and the fact that they overlap a little bit, it does lead to some sort of pathology overlap, but we'll talk about that in a bit. So this is a highly regulated enzyme, and the thing that activates this is this molecule called N-acetyl glutamate. The thing that we need to know about N-acetyl glutamate is that there's something else that cause its upregulation. So there's a process that upregulates an enzyme that will make more N-acetyl glutamate, and then that will activate carboxyl phosphate. So I'm just going to draw a little dotted arrow over here. I just need you to know that something downstream will affect this enzyme, and then this one is going to be the major regulatory step of this whole entire process. There is a deficiency over here. It's going to be CPS deficiency. And we'll talk about, well, I think that the best thing to do is to make a list or like a charts with all the diseases, and I'll talk about the symptoms that you actually get. But this is where the CPS deficiency occurs. All right, so the next step is where it starts to cycle, and the next step, none of the steps after this is going to be regulated, but what you need to know is some of the major enzymes. I'm going to draw in blue the enzymes that you have to know, and then if you don't need to know them, I'm going to draw them in black as enzymes, and then I'll make sure that all the enzymes you need to know are highlighted. Alright, so the next enzyme is called ornithine transcarbamylase carbamylase. And usually this is called OTC. And what really happens is there is this molecule called ornithine. And this is where the cycle actually occurs. So if you think about like before this part, it's not really a cycle. It's just adding substrate into the cycle. It's like the TCA, how you add acetyl-CoA and there's something that cycles constantly. Well, ornithine is one of those. It's almost like the equivalent of OAA. So ornithine comes in, combines with your carbonyl phosphates, and then you get this molecule called citrulline. Let me just move this around. And this citrulline then gets transported out to the cytosol. And it combines with a substrate that you know about already. It's aspartate. So that's where our two substrates come in. I want to just draw them in red because they're really important. It's where they are and where they enter. So your aspartate enters in from the cytosol, your ammonia enters in from the mitochondria. They combine. There's a enzyme that you don't really need to know. It makes arginosuccinate. It passes by another enzyme that you don't need to know, and it makes this amino acid called arginine. A side reaction that you need to know is that this actually makes a fumarate that can't be really used in the TCA, right? So yet because it's in the cytosol. So just a little side note: there's no TCA in this one. It has to be converted to something else before it goes back into the mitochondria. This arginine is the key molecule. So if you remember the structure of arginine, it kind of looks like it. The last thing has like a nitrogen, like this, and H2, and there's like another nitrogen like this, and the rest of the amino acids like that. Well, this over here looks really like urea, and that's exactly what happens. This arginine loses that group, and it gets rid of a urea. So urea comes from the breakdown of arginine, but all those nitrogens that culminated to that arginine is going to be your aspartate and your ammonia. Once that arginine loses that urea, it becomes an ornithine already. So this is also, this is actually another modified amino acid, just like taurine and stuff like that. But then your cycle continues on and on. I forgot to mention that there's one more deficiency over here. It's going to be called the OTC deficiency. But I'll see if I can get a chart out so that you guys realize that what type of symptoms you would get. All right, the last thing I need to talk about is going back to this over here, the N-acetylglutamate. There's one thing that will activate this this enzyme that will make N-acetylglutamate. So if you actually look at the name of it, it's N-acetyl glutamate. So the constituents is actually acetyl-CoA, and it's a glutamate. And there is an enzyme that we don't really need to know that catalyzes this enzyme, this reaction. But that enzyme is activated by arginine. And that's it for the Urea cycle.